Okay, welcome back, everybody. So we are going to continue with the after lunch session. It's the task for you to keep everybody awake.
I'll try.
Yeah, I'm sure you manage. So it's a pleasure to introduce you. Amrit Paudel as a scientific leader from the Research Center Pharmaceutical Engineering. I think you worked at the University of Graz, as I acknowledged. You worked for Bristol Myers Squibb. You were honored with the Young Investigator Award in the past, so also congratulations for this one. And you're going to introduce you on the topic Innovative Carrier Particle Engineering for Carrier-Based Dry Powder Inhalation. Thank you for this.
Thanks, Felix. And yeah, I hope I can make you awake, and I see some at least cups of coffees accompanying you, so you can choose. But of course, I will be talking on DPI engineering aspects with a main focus on carrier-based dry powder engineering, dry powder inhalers, and this is the agenda today. So we'll talk a lot about particles, particle engineering, design concepts, and also not only particle engineering on their own, but also downstream processing, something related to filling, like the topics that was also touched base this morning by the speakers. And also, while this journey, there's just a glimpse of some tool that might help in digital design and development of DPIs, and with some conclusion. So I would not spend much time in the introduction. This is again some of the redundant information from the morning. We know that the market in DPI is quite in local lung delivery with pulmonary inflammation as a major area, and some with infections. Majority small molecules, but also there are some, interests of biologics delivery to lung, from liquid formulations to DPIs are growing as well. So in addition to markets, there are quite some time exploratory research to maximize pulmonary drug delivery as a powder to really achieve regional deposition, targeting, and formulations. At least the intention of therapies are getting complex, more combinations, double, triple combinations. Drugs like just mentioned biologics, there are incentive vaccines and amorphous and all kind of this solid drug formulations. There are sometimes poorly soluble drug formulation, balancing act of like a long retention versus absorption. And also there's always incentive to decrease the frequency of treatment and so on. So anyway, so DPIs, as any other inhaled deliveries, is a combination of formulations, device, and patient that design or that needs to be part of the TPP, target profile, to design such product. And this is again information that you are very familiar with as you work in this field, most of you. There are carrier-based and carrier-free as a broad classification of dry powder inhaler. Carrier-free are the system which have, let's say, some minimum dispersant excipient with a major aerosolizable size APIs that are engineered to be delivered. So the majority in the market are carrier-based, where they are low-dose formulations containing low amount of drug of inhalable size, and as a simple concept, they are made to adhere onto the coarse carrier surface. Like always, lactose monohydrate is the gold standard. There are emerging other ones we'll discuss, and they are with the help of device, capsule-based, reservoir-based, multi-unit, single unit, and some dispersion mechanism helps them disperse to reach into the lung. So, carrier-based DPI, like a particulate formulation in general, but especially carrier-based DPI is a lot about surface, because you have these tiny inhalable size APIs that are adhered to the coarse particle at a surface. That are, like you see the size range difference. And when there is inhalation maneuver, you want them to detach and reach to the lung. So yes, there's a really need of balancing act, the opposite act of the interparticle strength between drug and excipient. They need to be strong to withstand processing, manufacturing, drug product process, flow, mixing, filling, etc. But also at the same time, they should be weak enough that can facilitate dispersion detachment. So as we mentioned, mostly since the major job is done by carriers, that's 90% and more than 90% mass fraction in your formulations, much more. So they are main drivers, their properties, their surface chemistry, surface topography, everything that are coming from surface properties are the main driver of enablers or innovation in DPIs. So talking about properties, we saw it's a delicate balance, and there are a lot of properties of them: mechanical properties, surface chemistry, surface interactions, surface energetics, and Daniela gave very nice case studies of tribo-charging of powders. So is quite a lot space of a degree of freedom, if you call particle passport to lung. And that's why it's sometimes called multi-particulate nightmares. So this is, I think, from Peter Stewart, named this many years back from Melbourne University. So with this introduction, now I will guide you or take you through some case studies of carrier particle engineering. That's the main focus. And what are emerging trend and some work that has been done from our group in past years. So overarching goal of designing carriers or in general particle engineering, but again, main focus is carrier is, of course, to improve drug delivery, the detachment performance, long deposition, regional deposition, and also enhance stability, that they have really repeated performance. A lot of time we know in our daily business is simply you can have same long deposition, but you want to just kill the variability. You don't have variability. So if you can really engineer, but also get same average performance, but they are less variable, that's a winner, both for the development, also for the patient, actually. And also, they are quite a remit. If you can really improve processability, drug product process by engineering, that's also the one drivers. So with this, I take you to a few case studies, of surface manipulation of lactose carrier, so in general, DPI carriers. And we start here with a case study where we applied weight decantation to remove surface fines. So basically, it sounds like a simple act in a way that if you think of inhalation grade lactose, commercial lactose, by definition, they come with some distribution. So they have fine fractions. So if as a formulator, you have to add fine fraction, typically you add as a dispersion aid. There's always a challenge to really calibrate, like how much fraction you want to add against what is already existing. So if you can simply clean those fines, but at the same time, if you can preserve their solid state, their morphology, their polymorph, that's quite an advantage. So this is something we started doing many years back, simply is a anti-solvent washing. So it's based on the principle that if this is through anti-solvent process, decantation, your fine particle dissolve and coarse particles simply settle down, and then you separate based on that. So here is the example. We started the commercial inhalation grade lactose monohydrate, tomahawk shaped crystals that we managed to clean quite a lot. So you can really see very smooth, fine free surface, still preserved as a lactose monohydrate, and it's crystal shape. So another approach is basically we let this lactose coarse particles being there, but then these fine particles is something we intend to develop as a bridge for coarse particles. So this become like a granules. So it's not really granules. So you start from solution in that way, but you really optimize like a spherical crystallization, with anti-solvent crystallization in a way that so you optimize this anti-solvent temperature process like ethanol versus acetone type to create the lactose carrier with different types of surface asperities, or let's say specific surface area that can help you control the drug loading and the reproducibility of the loaded small drugs. And the other one is, okay, we don't start with solution, now we start with suspension. So in a way that what we do is, in the earlier one I was showing was we were just cleaning by decantation. Here we apply the concept of surface tuning by optimizing the rate of surface dissolution versus surface crystal growth. So basically how it works is you have the suspension, you let it pass through this tubular reactor, crystallizer, that has two temperature gradients. So that's with a hot bath and cold bath. In the hot segment, you have dissolution, controlled dissolution, and in the cold section, you have the controlled growth of the crystals. So that way, by optimizing the residence time in these two parts, and then you can also add air as a plug flow to modulate the solution residence time in each part. That way, you can really have a controlled growth of the surface. So here, this is an example of mannitol. We start with this commercial mannitol of inhaled size range, and at the end we still create this beta lactose, beta mannitol, beta polymer. But what we also managed to create is the small growth of alpha mannitol at the surface of the beta mannitol, so that you can really manipulate specifically the surface roughness and surface crystal form of this carrier by cycling. So what does it mean? You know, these were all series of papers we have published for many years. These are techniques, but you see this is like a kind of a summary of this in vitro performance, NGI performance from these all carriers. Okay, so so this is a salbutamol sulfate. So I think in those papers there are also other couple of drugs, but this is like a compilation, just a case of salbutamol sulfate fine particle fractions. So you can see this Inhalac 70 untreated versus weight decanted. You already gain better fine particle fraction by 12% increase. Then in this case, when you have the spherical agglomeration or spherical crystallization, you can get in one case basically the one with the acetone-based crystallization, you really significantly increase FPF, while with the ethanol-based crystallization, where you have big pores, basically you can load a lot of fraction of fines, the drugs, but they are not coming out. So basically you reduce there. And in another case, the one I was showing, the crystallization cycling, again from there you can really achieve quite an increase in fine particle fractions. So by changing the surface topography, right? So and here is the specific surface area. You see it's not only the bulk surface area that you can classically measure by BET or gas phase substance. Is really specific tuning of surface topography that led to such behavior. One example here is out of those is if you just see the work of adhesion of alpha versus beta polymorph of mannitol. So beta has quite a strong adhesion force compared to the alpha. So if you let alpha crystal growing on the top of the surface of beta, you can really tune, based on the surface coverage of beta, the strength of adhesion of drug onto the surface. The second case is, again, now here is a lactose carrier, but instead of surface topography only, here we are looking into the change in chemical composition, so the anomeric composition, but also the shape. So, you know, like with the lactose, if you just see crystal packing, lactose shape is quite inherently related to its molecules. If you have lactose monohydrate, if you let it crystallize, it automatically crystallizes this tomahawk shape crystal. But then you can also do those engineerings that I was showing before. So you can really have alpha lactose spherical here, but also in this case just anhydrous beta lactose, which is again non-spherical, some acicular shape or plate shape. And this is the spherical lactose obtained by spray drying So where you have really like around 50-50 composition of alpha and beta enantiomer. And what you see is, like irrespective of enantiomeric composition. So this is again FPF of salbutamol. So if you just compare this bar and that bar, they are both spherical, but with different enantiomeric composition. So here, basically chemistry, so chemical composition of lactose doesn't matter, which makes sense because it's a surface phenomenon. So it's mainly this spherical is much less better or worse than the shape. So here, now if you look into this one, so this is Tomahawk alpha monohydrate, lactose monohydrate is a beta. So beta has some intrinsically higher FPF, but in general, if you have non-circular lactose, irrespective of chemical composition, enantiomeric composition, you are better with the FPF. So now the another case is basically we took this time with a mannitol instead of lactose, and we compared two techniques. So basically one is this wet sieving. So wet sieving in a way that the earlier one we were showing decantation was to remove fines. Here the way to do wet sieving is really, so you control, so basically you have a solvent and you just do the sieving process. And they use really select solvent in a way that you can have control erosion of the surface. So in a way that you create such surface. In other case, basically it's like this is spray congealing. This is something you cannot do with the lactose because lactose degrade when it melts, which is not the case of mannitol. So mannitol melts are pretty stable until the range you can atomize. So like melting plus 20-30 degree. So basically we atomize the melt to create very spherical particles as a carrier. Now you see here, this is the weight sieved particle where you create like this kind of asperity, very controlled asperity. So basically you through the erosion of the surface, still beta mannitol with the roughness, quite irregular surface. While the spray congealing, again, so with spray congealing, there are certain fines, but if you see majority of them with a very nice, smooth, spherical surface. And in this case, again, this was like a alpha mannitol predominantly with some trace amount of delta crystal at the surface. But they are very spherical, unique crystalline solid of mannitol. So what does it mean? So here now, this is a FWF, the fine particle fraction, and on the top is a short weight consistency of different carriers, the spray congealed versus wet sieved, and in this case is a beclomethasone dipropionate, 1% as a binary and ternary agent. So what you see in general, so if you just see here, and these names are here, I don't know whether you can read. So by making spray congealing, so if you make spherical, nice surface of mannitol, even without ternary agent, you are very good with a short weight consistency. So that acts the best actually. While for others there are variability. But now if you look into FPF, so basically compared to binary blends, if you just see by wet sieving, if you see just binary blend untreated, spray congealing already like double the fine particle fractions, and also wet sieving with the pre-blend significantly increase the FPF. So it's really a combined effect you can achieve. The third case study is I'm giving here is the impact of added fines. So we know that we use lactose fines on the top of coarse lactose to, in a way, control the dispersion. So there are a lot of theories. It works, but mechanistically not all proven. So you can passivate the active surface per se, so-called, and so on. But anyway, what we are looking into is, so if you are developing that as a strategy, so obviously if you have fine materials on your sample, and you can imagine they have higher active surface area, and then you can have also the consequence on the stability. So what we did is we look into, these are two commercial budesonide DPIs with same drug loading, but only the difference is in one case, with very high fine particles of lactose, for lactose fine. So those below 10 micron were more than like around 10% mass fraction. Whereas in the second commercial product, it's almost like 0.5% of that. And what you see here is the deposited fractions, and then what was done is that they were stored at very like a provoking condition. For example, like when we talk about the patient, they forget it in the bathroom, let's say. So you can call it like that. And you can, and this is also already published work, by the way. You can see obviously the one which was with very high fine, there's a significant increase in the MMAD size. So the size grows significantly, and that is boiling down into the very high extrathoracic deposition. So there's a loss of aerosolization compared to the one where obviously there's very less fine, and MMAD remain pretty constant, and also it still behave pretty well in terms of in vitro deposition over 28 days. The second case is, so that's now looking into that. So these were commercial product case. And what we did is we made our own DPI with budesonide again, but there compared to binary system, this is a control black, then we added different amount of fines. So let's say this is 5%, A, and the C is 10%. So just for a number. You can see the sensitivity of humidity, for example, for the intermediate fine content doesn't change that much, so it's pretty much remaining same. The one with the higher fraction, now if you expose to like a room temperature 75% humidity, you have sharp drop in the in vitro deposition. And what is something that not that much appreciated is dissolution. So imagine you have, this is a dispersion. So now what we did is we isolated with classical Copley cup in NGI this deposited fraction, and we just run the dissolution using simulated lung fluid of the drug budesonide from these formulations. What do you see is the binary system shows this black type of dissolution profile, and until 5% fine, it doesn't change. And if you have more fine, like around 10%, now you see notable drop in dissolution profile. So basically, there's a liability of dissolution of budesonide like poorly soluble drug crystals when you add too much fines on that. So other than that, like there are fines, there are different ways to manipulate, but also you can really engineer this roughness and those surface topography by simply working on the transition chemistry. So basically, if you let this smooth spray congealed mannitol anneal over time, you can create such rough surface simply by annealing, because now you have really this delta polymorph transforming to alpha at the surface that create very controlled topography on the surface. So that's also a very smart way to engineer. So really work on the kinetics of annealing to create the intended roughness of the surface. Something we're also working is, like using the inkjet printing as a principle to create particles, carrier particles. And there we can use really the substrate with known roughness. So you really have, you let these droplets deposited or those crystallizing into this known rough surface to counter engineer the roughness of the particles. So those are some exploratory particle engineering innovations for the carriers. Now you engineer API, you have adhesive mixings, and I mean there are also, a very important part is also downstream processing of DPIs. So we look here, what are the consequences of carrier particle engineering, or in this case, drug particle engineering and downstream processing. So this work also already published one where here we looked into two types of salbutamol sulfate. This is spray dried. You see spherical particles, jet milled, classical crystalline needle type of material, while spray dried was predominantly amorphous. We made adhesive mixtures with two different composition, like 5%, 2% and 5% loading of drug. You can see how the shape on the one hand and also the loading on the other hand change the tribo-charging behavior of the blend actually. So this is what I was just saying this morning, like when you have individual material, drugs, tribo-charging that Daniel was showing, and the excipient, when you mix, you create different structures. And that can be manipulated by, obviously it has the fines loading has an effect, but also the way you engineer fine of drug can impact that. So other thing not directly related to particle engineering is also in some direction of tribo-charging. Here we're looking into different types of inhaled capsules, different lubrication profile, different filling speed. You can see how the tribo-charging profile changes based on them. So this is a combination of humidity, like filling speed, as well as the capsule types itself change all these tribo-charging behavior. And again, here, this is the last case where we are looking into the impact of carrier engineering on the filling profile, the filling quality. So basically, fill weight and fill weight variability. Again, here we look into different settings. So setting 1, setting 2, this is a dosator-based filling. And it's nothing but the compression strength, basically. So how far from the bottom of the powder bed your dosator is. And here, what you see typically is when you engineer carrier, so lactose engineered carrier is more resilient to the change in process parameters. So it gives more process robustness compared to the unengineered crystals. So these were those, the one I was showing in the beginning, this decanted, weight decanted lactose, where you have fines removed there. So they are more controllable in terms of even capsule filling quality compared to the one with the unknown amount of fines and surface. So yeah, with that, while doing that, we also have developed some inline monitoring tool in the capsule filling bed to look into the segregation that can happen during capsule filling. So, so using this inline Raman, so this, you see these continuous lines are Raman's based quantification of drug in the powder bed during filling, and these dotted lines are those measured by HPLC. And due to this, what you can find, so with this we use, so basically, here you see the segregation, and we introduce some mixing element here, like a physical mixing element that can quite improve the mixing homogeneity. The other inline tool was based on terahertz spectroscopy to monitor the relative density during the filling process. So that's already giving you, based on the particle, like the blend density, you can predict the fill weight then you could achieve. So I'm already at the end of my presentation. So I think, these are all experimental use case, but like we are talking about material innovations. So there are a lot of things you can learn from theory, from models, all kind of atomistic models, molecular models, to process models like a CFD-DEM or other mesoscopic model, and connect these properties of particles to the performance. So this was like many years, this European cost action, the prediction of inhaled products performance, which we are also part of. And I just give you one example, again, related to particle tribo-charging from the morning. So what we try to do is just using this at DFT calculation to calculate the parameters, and this is this ionization potential in y-axis, and make their comparison with the experimental tribo-charging. So this is specific charge density, nanocoulomb per meter square, experimentally measured in Faraday Pell. And what happens is, like what you were showing this morning's work function, this is something very difficult to measure. And also it's very difficult to calculate. You have always inaccuracy. But this work function of any material is always related to its ionization potential and this Fermi level of energy. So if you imagine if this is a constant, actually work function is quite related to ionization potential. And this is something you can measure better. And here this is, like even this gas phase molecular property can really help early on. What would be the level of tribo-charging you can expect for the certain type of crystals. Another type of modeling is, like this is MD simulations of drug crystal over the carrier crystal. This is lactose with a lot of drugs, and you're just measuring the energy. So, attachment energy as a function of distance, and that is giving you insight into the strength of interaction you can expect on the crystal surface. Yeah, so with that, so these few examples, I already am summarizing that, like in DPI particle engineering, today I showed more the case of carrier engineering. So there are a lot of possibilities. So particle engineering doesn't mean only crystallization or just spray drying. So there are other possibilities that can be explored and maybe smartly implemented. Just we need to understand what do you want to achieve with that, and also we should appreciate the possibility that we can innovate using that. Downstream processing, I just saw some case studies, at least, I mean, that's in research setting. This is something that's not much done. And I know PAT tools can really help there. And the final one is this digital design is way to go. You can really implement to design things. So basically, I'm acknowledging some of these scientists that contributed from my group, the Austrian Funding Agency, and a lot of industrial partners. They work with us over the years to achieve that. And thank you for your attention.
Thank you. Time for questions.
Yes, Felix.
Yeah, I have one question. You mentioned dissolution testing at least once with a recent publication.
Yeah, no problem.
From your perspective, can you explain why there have been differences driven by the powder? What do you assume?
Yes, so actually, well, in this particular paper, we didn't explain, we just did it. But what we know is, like this, I think this is the guy in University of Bristol, Professor Reeds, Jonathan Reeds, is quite famous also in inhalation community. He's doing some very interesting experiments sometimes. What he does is, he has this levitator, tweezer levitator, because you cannot do with ultrasonic one. And he monitor, evaporation with MDI, but also how aerosol size sugars are interacting with moisture. And what is noticed is, like when they are very small size and you're exposed to very high water activity in the beginning, so these sugars take quite a high amount of water, and it really makes like a gel surface, actually glass surface, after which the diffusion of remaining moisture is significantly decreased after that. So that's our expectation. We have not proven it, but he has proven that, not in dissolution setting. So, like if you have these poorly soluble crystals and there are a lot of filthy lactose around, let's imagine. So until some, like some time point, it takes a lot of water. And after that, basically you also see in our profile, so initial rate are quite similar. And after some time in this inflection, the extent it decreases, and that's something we are attributing. So basically, the lactose is already forming, in this, because this is in vitro, so it's in thin sandwich type of setup. Try to mimic, not like a, not in vivo, but anyway, it's not like a paddle basket. So we think that is discriminating the situation that you form this glassy layer of lactose around the budesonide crystal. Plausible explanation. Yes, please.
Thank you. You briefly mentioned PET tool, Raman spectroscopy to monitor the blend homogeneity. Is that correct?
Yes, or other way around. Yeah, it's a segregation tendency.
Yeah. Can you just spend a few words about the limitation of this PET technology?
That's very interesting. So, in which context you are saying? Is there like a limit of?
It's the penetration depth, for instance, into the blend itself.
Yeah. I mean, I think it would have such limitations, but like in this case, we are just looking into the layer of the powders, which is
just the top surface, right?
Top surface, but just considering that. So the depth is, so it's not like very compact tablets or something. So you have a layer on the surface, and actually this is like a classical fat probe. Like this the 794 nanometer wavelength, and at least the beauty is sensitivity is less compared to the strong laser, but you don't also have the what you call fluorescence interference. So that helps you gain more, let's say. Specificity in this case.
Thank you.
Yeah. So, and what you have seen is basically those all dots were actually HPLC measured concentrations, and the average were the Raman ones.
Regarding that graphing.
Yeah. Can I go there? How can I go there? Or,
Just
yeah, because there are a lot of interest there.
I don't know if I understood it correctly. So was
Yeah.
this during capsule filling that
I can help
had?
you understand. Don't worry. So, which one? Wait, I think that's before that, right? Here. Yeah. Tell me. So this one.
Yeah.
So this is basically in the capsule filler. So this is the concentration in that is in the powder bed in the filling machine.
And so I'm curious, how did you conducted then that mixing step?
Okay. So that, I mean, I recommend you to read this paper. Basically, so first we found this. Okay. So this was part of project where we were modeling the powder bed. So basically, based on this, we now on that surface, because what is happening is you have this dosator process. It's making powder whole and this dynamics, right? So, and we did some modeling based on this experimental data, and we have introduced some mixing elements. It's like hockey stick kind of things. That's scrapping and mixing. Okay. So I didn't mention it here. Actually, in my previous version of slide, there was some picture here, like the introduction point.
Yeah, I was curious about that because I was wondering if you stopped the process, mixed, and
no.
So it was an inline mixing.
Inline.
Oh,
so it's
okay.
like hockey stick, so it's kind of scrapper. But this was done based on this finding and some modeling, designing that tool to add that.
Thank you. Thank you very much.
Thanks a lot.